Um, so welcome everyone to today's session. Let me just share my screen. Okay, there we are. Okay. So, there's my So today we are going to talk about phylogenetic trees in public health. Of course, I do realize that we are a mixed group, so some of these might, be familiar to some folks, but for some it might be, new information. But I can hope that this is useful to all of you. Especially remember the main um, aim of this course was the RC typing workflow that does generate a phylogenetic tree for which you can use for different or for that I can apply to different users in public health. So the idea is to also go into a bit more onto the phylogenetic trees. So, of course, here, we start with what is epidemiology, um, because, this is quite critical to public health, which is the study of distribution and determinants of health-related outcome in a specified population and the application of that study to control health problems. So ideally thinking about who, what, when, where, uh, within, different populations. Now, genomic epidemiology, for which phylogenetic inference applies, now is the use of genomic data to understand the distribution and these determinants of health-related outcomes. So, whereas in general epidemiology you have different sources of data, for genomic epidemiology your source of data there be pathogen genomic data. So genomic epidemiology does comprise two things. So surveillance and typing of pathogens over time and space. So similar to what we did with Theiagen. So you do surveillance and then you get genomic data out of it and it gets some typing done. And then you look at the evolutionary history of these pathogens. And genomic epidemiology does facilitate three broad things. So one is the identification of introductions of new pathogens. And the kind of questions you're asking here is, what is this new pathogen? What is the reservoir? Is it human? Is it an unhuman reservoir in this case? Two, we want to understand the transmission dynamics. Again, here you can ask different questions, whether cases are linked together. Is there human to human transmission, or is mostly jumps from a non-human to a human? When was this pathogen introduced in the population of interest? And also the location at which it was introduced. Lastly, understanding the pathogen evolution. So here you're asking yourself, is the pathogen changing? And if it is, what is the rate of evolution? Where these changes occurring? And there are also like unique changes for different populations. So having had that background into genomic epidemiology and the kind of questions that genomic epidemiology facilitates, we want to introduce ourselves to phylogenetics. So what is phylogenetics? Phylogenetics is the estimation of ancestral evolutionary relationship among pathogens, um, and using different sources of data. So we could use sequences from genes, genomes, or even proteins. And the basic principle here is that more similar sequences will be closely clustered together and are closely related, whereas, pathogens that are quite divergent will cluster further apart. And once you establish these evolutionary relationships, these are typically visualized in the form of a phylogenetic tree. Again, because we have folks here who work with bacteria, typically here what we are looking at is that vertical history rather than the horizontal transfer or horizontal relationships between pathogens. So we are looking at so thinking, going back so that you can establish the most recent common ancestor of a given pathogen, rather than, for example, where a resistant gene has been shared to another organism horizontally. And in such case, you'll have more of like what you'd call convergent evolution rather than an actual evolutionary relationship. So a phylogenetic tree is a diagram that is used to depict these evolutionary relationships. As you can see, this phylogenetic representation of, sort of what is thought to be the first representation of phylogenetic tree by Charles Darwin. Again, showing which genes or organisms are closely related and which ones are more distant. And if the tree is rooted, we'll look at the concept of rooting. You can be able to understand the direction of that evolutionary process. So here is a rather simplistic representation of a phylogenetic tree. So we have what we call leaves or taxa or tips. Again, they're represented differently in literature. And in this case, these leaves or tips will be A, B, C, D, E, and F. And then we have nodes. So these would be the most recent common ancestors or the ancestors to the existing pathogens or sequences that we have. And then we have branches that, again, we'll talk about these in more detail that represent the nature of the evolutionary relationship between the sequences that we're looking at. And again, like I said, if the tree is rooted, you can look at the concept of time and directionality. Again, seeing movement from left to right here, ancestors to the descendants. So in an outbreak setting or in an outbreak investigation setting, looking at the evolutionary relationship is a good proxy for the likelihood of transmission. So because, in an outbreak, you're trying to understand who has infected whom. And of course, by estimating the evolutionary relationships amongst your samples, you are trying to establish the likelihood of transmission. So sequencing and bioinformatics methods offer this high resolution means of understanding the genetic relatedness. So with that background, the next thing we want to look at is how do you build a phylogenetic tree? Again, if I'm moving too fast and if you have a question at any given point, please do stop me and ask the question. Happy to go over some of these. I might be going a bit fast because there's a lot to cover, but certainly it's better to have a better understanding than cover a lot and have little understanding of it. So now that we understand what a phylogenetic tree is, how do we build them? So the general approach to building phylogenetic tree encompasses 3 steps. So one, or rather the first step is identifying the genetic differences among the samples or the sequences within a data set. 2, typically involve removing any differences that are unlikely to come from ancestral relationship. And 3 is using the identified differences to infer evolutionary relationships. So we're going to go through each of these steps and then look at what each entails. So to identify genetic differences, there are multiple ways in which you can do this. So on the right here, we have, again, A, B, C, D, E. So these are the taxa or the types of the sequences that we want to infer evolutionary relationships amongst. So you could look at single nucleotide polymorphisms. So KMAS, here you are identifying nucleotide variations between the samples of the sequences. And you could look at much larger stretches of genetic differences. So where you look at either core gene or whole genome and also multi-local sequence types. So here you're looking at allelic variations rather than identifying these single nucleotide variations. The second step, typically optional, but depends again on the pathogen and also the tool that you're using, is to be able to mask differences that are likely to result from ancestral relationships. So one such thing to consider is recombination, for example. So recombination is known to actually interfere with the building of phylogenetic tree or establishing true ancestral relationships between samples. And if you do have sequences or regions within your sequences that recombine, it's often advised to remove those. Secondly, also you might want to remove segments that are likely to be misaligned. So this could be duplications or rather repetitive regions that might be mapped randomly in, depending on where they are repeated across the genome. And, you could infer or make wrong inferences just depending on how the alignment was done and where some of these repeats were aligned relative to the other sequences. And also thinking about accessory regions of the genome that either might not be inherited vertically or might not be present in other sequences in an evolutionary manner, but so then they might actually skew your inference of evolutionary relationships. So whereas this is marked here as optional, so these are actually things that you might put at the back of your mind depending on the pathogen that you're working with, and be able to check for specifically for the pathogen that you're working with. All right, the third step is actually the inference process. So again, here is a rather simplistic way of looking at this, but it does explain the process quite efficiently. So if you think about on the left here, we have a representation of an alignment, again, with the associated mutation. And on the right here, we have an inferred phylogenetic tree from it. So using the simplest model here, you can see that, all the sequences, they at the root here or the most recent common ancestor, you have the blue because, all the sequences have the blue. And then some of these have the orange, so that's A, B, and C. Again, this will be at the most recent common ancestor of those three. And then A and B do share this mutation that is green. But, because neither A nor B has an additional mutation that differentiates the two, you can see that, it's a flat or it's, vertical line there. But C also has the red mutation that differentiates it from that branch that leads to A and B. And of course, D and E do share this teal mutation or cyan, if I got that color correctly. So you can see that they share that most recent common ancestor. But then they also have this red mutation, which though shared, same color with this one, is at a different position from this mutation that C has. So red, so that could actually indicate like sort of the same amino acid mutation, but again, at a different position in the genome compared to what C has here. So typically, the inference process uses complex mathematical model to predict how likely the sequences evolved. But I believe that this suffices to just simplistically explain how that happens. However, there are typically, important problems to think about in this when you're trying to look at the molecular evolutionary process of pathogen. the first question is whether there is any tree at all that is, because recombination, if it happens too much, then, you might not even be able to build a tree because that will affect the tree inference process. Building a tree is dependent on the number of tips that you have. There are many possible trees that you can have. Typically, whatever the software gives you is the most likely one. Does not necessarily mean that it's the only tree, or it could be the one with the highest likelihood, depending on the method that you're using. And as you can see here, for 10 taxa, there are 2 times 10 to the power of 6 unrooted trees, and you can see how that rises exponentially. When you have 50 taxa, you have 3 times 10 to the power of 74 unrooted trees. However, they are efficient and powerful such algorithms that have been implemented by different tools that is able to quickly sort out through the number of possible trees. Again, most of these methods don't actually go through all these possible trees, but they have efficient algorithms that are implemented to only evaluate the most likely trees amongst the possible ones. So if you think about, the tree building process, there are sort of like two major different classes. So one is those that do not employ an explicit model of sequence evolution. So these are thought to be the very simple way of evaluating these evolutionary relationships. And they do apply the parsimony principle, the simplest or the fewest number of mutations explains the tree. And on the right, you have the methods that do apply explicit models of sequence evolution. And here we can have those that do pairwise comparison of sequences, for example, the distance-based methods, neighbor joining, for example, or AppGamma. And you have the statistical approaches, those that employ the maximum likelihood method. So this will be typically in a lot of the tools that we've used. And those that employ Bayesian methods, the common one being BEAST, if you've ever used BEAST. So again, different methods. And the question, is which one should you use? We'll see that in a slide. And in addition to the different methods of building phylogenetic trees, you also have to think about the model that actually explains that evolutionary process. So there are different models, and these allow for the correction of genetic distances, or what we typically refer to as the unseen substitution. So yes, you do have your alignment that has these A, T, Gs, and Cs, but again, you have to think of the evolutionary process that led to what you're currently observing. So there are different models that try to define these evolutionary processes. They do consider, for example, the relative frequencies of the 4 nucleotides. Some models, for example, will assume that you have an equal number of the nucleotides, so each has a proportion of 25%. Some will, for example, assign different frequencies, for example, to the purines vis-a-vis the pyrimidines, or like, for example, depending on the GC content of the pathogen that you're looking at. Then there are also, considerations with regards to relative rates of substitutions between the nucleotides themselves. So as you'll expect, a change from between nucleotides of the same type. So let's say purines, for example, will have a different relative frequency, relative rate of change compared to between a purine and a pyrimidine. Okay. So for example, here in the first model, this is assuming that changes to any nucleotide is relatively the same. Here, change to T to C and C to T is the same. A to G and G to A is the same, but T to A is different. So is C to G. And then, you have sort of like what you'd call the general time reversible model that, change to any nucleotide has its own separate frequency. So again, from simple to very complex models, here is a list of some of those, like the Duke Cantor 69, Pfaffenstein 81, Kimura 2-parameter, HKY to the general time reversible parameter. So different models that you typically have to choose from. And also, as you will expect, different tools have different models on default. So some of the decisions you need to make as you're running your phylogenetic analysis is one, of course, there are many ways to make phylogenetic trees. So which method are you going to use? Which substitution models are you going to use? And the most appropriate way or method or model that you're going to use will depend on the organism that you're looking at, the data set, as well as the purpose of your analysis. For example, if you just want to have a general look into the data set, you will most likely look at a method that employs the fastest algorithm. Might not be accurate, but remember, your whole objective is just to look at the data quickly. So at that point, you're really not looking for the most accurate method, because then again, typically there's, this trade-off between speed and accuracy, whereby the most accurate method takes time. I've run beast analysis that takes a month or more. But sometimes, you might want to just have a quick run that takes a few seconds to a minute to actually just give you a phylogenetic tree. So it's always advised to check, for that particular pathogen that you want to build a tree for, which kind of tools do they use, which kind of models do they implement. Again, for the data set, is this for research purposes? Is this for outbreak analysis? So such things, do you have data on time? Do you have some, epidemiological data that goes along with the genomic data? So again, the decision will depend on a lot of things that you need to decide upon. And lastly, something that I've emphasized before is sample selection is quite critical. It's almost like it's what determines everything. So always ensure that your sample set is of high quality, so they pass QC threshold. And we said, whereas there are no hard-coded QC threshold for every pathogen, just ensure that do they pass QC threshold that has been set either for that pathogen or like the thresholds actually are appropriate, like do they meet what is expected of that particular pathogen um, uh, for it for you to qualify that as a good sample to undertake downstream analysis for. And you need samples that can answer the question that you want to address. For outbreak analysis, samples should be taxonomically related, so they should be the same species, usually the same sequence type or lineage, depending on how you're characterizing your sample. And just be aware that, typically, you don't want to bundle sequences, because again, you're looking at genomic uh, um, or the evolutionary relationships which come from the genomic information. So serotype can be a slightly different concept. So we've seen this scenario where um, you can have samples or sequences that are the same serotype, but definitely different sequence type, because again, serology and genotype, or rather phenotype vis-a-vis genotype, are quite different things. Or, they have the same AMR profile, same resistant genes, but, this could have been inherited horizontally rather than vertically. So again, think quite critically about the selection of samples that you want to undertake phylogenetic analysis for. So we've built the tree. Hopefully, you've decided which method you're using, and you've run your tree similar to what we did with RC typing workflow. You have a tree. How do you interpret the phylogenetic tree? I intend to go over different ways of visualizing the tree in the office hour on Thursday. But if there is time today, I can also just go over that. But for now, I wanted to just keep it more on the lecture rather than looking at the individual ways of visualizing these trees. So we've seen the structure of a phylogenetic tree. We have what we call the terminal nodes or tips or leaves, depending on different terminologies. So these are the sequence samples that are represented at the outermost part of the tree. That's why we call them the tips of the nodes or our terminal nodes. So this would be A, B, C, D, and E. And then we have the internal nodes. So these are the inferred sequences. So these we don't have sequences for, but are inferred from the evolutionary relationships between the terminal nodes. And these would be these ones that I highlighted in yellow here. And then we do have the branch lengths. And the branch lengths visual representation of the evolutionary distance or the number of phylogenetic differences. Again, in some trees, this can be as simple as single nucleotide polymorphisms. In some trees, especially if they're time trees, so the branch length might not just represent evolutionary distance, but also time component. So it's good to be aware of how the tree was built and which information was taken into consideration when building that tree. And in this case, because A and B have no differences between them, in this case, the branch length is 0. But then, we have a large distance or differences between A and C. And if you look at the branch length between D and E, that is much shorter than what we have between A and C. And of course, you can say that that distance between A and C is the same as what you have between B and C, because A and B have 0 genetic distance between them. Again, just reminding you that the differences are on the horizontal lines. So the branch lengths are here, and the vertical distance are merely for visual representation and actually don't mean anything. So this here can be as large as it can be. If I can just get my annotation here. So this can, you can expand it as high as you want or collapse it as short as you want, but most of that is just for visual representation and really is not useful for estimating the genetic distance. So clear electrons, close. And then we commonly, have the terminology such as cluster, referring to a group of sample with close evolutionary relationship. And of course, A and B can make up a cluster. D and E can also make up a cluster. And in some cases, again, depending on trying to, why is it? Oh, okay, sorry, my mouse. Again, depending on what you're looking at, A, B, and C can also form its own cluster. So again, the resolution of what defines a cluster depends on what it is exactly you're looking at. Okay. And some of these clusters, could form a lineage. Some of these clusters could be could refer to a clade, but remember the cluster can be used loosely to refer to a group of samples with a close evolutionary relationship. We also want to talk about S&P distances because this is commonly used in public health, especially when looking at or trying to delineate transmission between individuals. So these are the number of positions where the nucleotide varies between two samples. And often, remember, these do not use the evolutionary model of information. So this typically uses the actual number of differences that I estimated there. So remember the left arm of those two different classes that we looked at. So think of it more as parsimony method. So these are typically calculated from non-alignment used to build the phylogenetic tree. And if using the core genome tree, it calculates the core genome S&P distances. It can be used to define transmission classes if suitable S&P distance thresholds are available. But typically, what you say is that, phylogenetic and S&P distances are more not to define transmission, but rule out transmission events. So you're using phylogenetics to say that it's unlikely that, these two are related, but it's not mostly like, yes, definitively to tell you that these two sequences, there was a transmission event between X and Y. It's difficult to confidently conclude that transmission occurred between two people, places based on just phylogenetic distances alone. So typically you need some contextual information, for example, epidemiological data. You need to think about the unsampled population because the tree simply represents what you are able to sequence. What about what you're not able to sequence? So you need to think about that as well. For example, are you only picking samples from symptomatic cases? What about asymptomatic cases that also have been infected with that particular pathogen? What about S&P distance thresholds? So, again, there are a few things you need to think about. So some of these have been defined by different organizations, for example, PulseNet for enteric pathogens involved in food outbreaks. Defined in literature for some taxa, for example, TB or Klebsiella. But then, thresholds are very difficult to define between samples. Certain samples, again, if these are not predefined, because this will also vary depending on a couple of things. One, the time of collection between the samples. So you do not expect to rule out transmission between samples taken, within a month, or, let's say you had a study that, has been looking at pathogen transmission over a 3-year period. So you're going to have different S&P distances to be able to delineate those. So always take into consideration the time between those collection dates. Also, you have to think about the mutation rate. So some pathogens have very low mutation rate, probably like a single mutation across the whole genome in a given year, whereas some will have several hundreds of mutations across the pathogen. So for example, viruses have smaller genomes but high evolutionary rates. Bacteria, again, some have quite large genomes but very low evolutionary rates. So it's not just the genome size that determines that, but generally the mutation rates. And some of these will have been calculated prior, so you can get this from literature. The diversity of the pathogen population in these samples. So again, all these are sort of like connected. So you're thinking about the within host diversity. So what do you see when you pick out a sample or a colony from the same host? What sort of distance do you expect in a sample when it's evolving within a single host vis-a-vis between host. So obviously, the minimum SNPs within host evolution. So the maximum within host diversity of SNP should sort of be like probably the minimum SNP that you can use for between host transmission events. So I'll move to the next section. So routing a phylogenetic tree. So typically trees will be built either rooted or unrooted. But like we said earlier, rooting a phylogenetic tree gives it that directionality. You can look a bit more onto like, the ancestors and the descendants. To root a tree, you can either use an outgroup or you can do a midpoint rooting. So an outgroup here, for example, F, which is a sequence that does not belong to the in-group. So assuming here A through to E is the in-group. So it should be distantly related to the taxa, but not again too distantly related. So it should be homologous to the in-group. Or you could use the midpoint, which should be the middle distance between the two most diverged sequences in your sample set. Again, knowing how, whether the tool that you're using gives you an unrooted tree or a rooted tree, you'd always want to know how that tree was rooted. Was it midpoint, or is it typically done by the reference, or do they pick the most distant sample within your data set and route it for the building process of your phylogenetic tree. So the same tree, if you don't root it, so we have the root node K that disappears here. So we have an unrooted tree. So here we can't really visualize.
James, can I have a question here?
Yes, please.
Can you go back to the midpoint? The midpoint routing. So that would be at K?
No. So it would be at K, but my tree is not properly visualized such that you would say that the midpoint is exactly K. I would say the representation that I have here is more for having an outgroup F. But you see, because my tree, like you can see, basically all the branch lengths lead to the same point. So this is rather like a different kind of tree than one I would expect, because the midpoint could probably even be between H and G here.
So between J and H.
Or between J and H, but we don't know that for sure, because again, it depends. It's easier to figure that out when you have, so what I mean is, like, if I was to take this, and let's say if I had, let's even say A there, sorry for my very bad drawing. Let's say the C there, we don't know like where this could be. Essentially, the midpoint routing could even be here, like where we have D and E, but we don't know that for sure. It will depend on because typically you're looking at the two most dissimilar taxas, which could be A and let's say E or something, and then you find where that midpoint is, and then you route it that. I don't know if that helps, but it's hard to predict because this is more of like a cladogram rather than a phylogenetic tree. Did I answer
So
your
it's
question?
usually not that easy to. Can you hear me, James?
Yes, I can hear you.
Okay, so it's hard to calculate where the midpoint would be based on the unrooted or the tree as it is right now, and you.
So if somebody gave you a tree like this, it will be difficult for you to just visually like pinpoint where that is. Again, this looks simple because we just have what 1, 2, 3, 4, 5, 6 steps, right? But think of it like where you have 100, right? Obviously, it's going to have clusters everywhere, and of course, the software is quick to find those two most dissimilar taxa.
Sorry,
and,
James.
and put.
So I was about saying that. I mean, like you took the words exactly out of my mouth. If you use a program like Fig Tree that visualizes a tree, if you have an unrooted tree, it probably will just calculate that for you. You have 1,000 leaf nodes, and then to just tell you this is the midpoint. So it's not something I think you worry about.
I know that usually the tree viewer can do it for you. I'm just trying to predict where it would be. So the short answer is it's not easy. Okay, thank you.
Okay.
Yeah.
Okay.
Yeah.
Okay.
Thank you. Thank you.
Yeah. Thank you for the question. Yes, it's not easy. Well, I do, I would imagine in the olden days when we didn't have all the clever software or algorithms that we have now, probably people used to do this by hand. But yeah, again, probably those were fewer samples like we have right now. So yeah, this can be quite a task. Okay, so the other concept that I wanted to bring up was bootstrapping. So bootstrapping being a statistical.
Sorry, James, can we go back to the unrooted tree? I have another question. So
yes, please.
What would be unrooted trees good for? So when we would better use the unrooted view of the tree rather than the rooted one?
So unrooted tree mostly is focused on, like, as the slide says, you're interested in looking at the evolutionary relationship. I find them much better in understanding, like, or delineating clusters better than where you have a midpoint routing, because midpoint routing sometimes can force you to view the tree in a certain way.
Okay. So the unrooted one would be less biased when you are deciding on clusters or clades.
Yes. Yes, when you're looking at particular clusters, definitely unrooted is usually better. But when you're really interested in the directionality of the evolution, like we came from this, then to this, and then to that, then an unrooted tree will not tell you that, because everything like is everywhere. Yes.
Right. So usually to decide on clusters would better look at the unrooted tree, not the rooted one, because that even if we can see some clustering in the rooted tree, it might not be the real thing.
My personal advice is before
hello, James.
probably, yes, my personal advice would be do not or what I typically do is generate multiple trees. So before I come to my conclusion, I would look at the tree in multiple ways. Like if I root it with like a reference, if I midpoint root it, if I just look at the unrooted tree, what do I get? Like, I mean, what sort of communication, or what am I communicating? And there are also things to do with, like, if we even go beyond the distinction between whether the tree is rooted or not, like, do you have a rectangular tree? Do you have a circular tree? Do you have a radiating tree? Like, like, what is it that you actually want the person that you're communicating to?
Right, but then the circular or the rectangular tree, it's basically the same thing, just like viewed differently. But I mean, the relationships would still be the same, regardless of whether you are looking at a rectangular or a circular one.
But you will agree with me that sometimes, depending on how somebody presents the circular tree vis-a-vis the rectangular tree.
Right, sometimes it's easier to look at one of them than the other.
Exactly.
Right.
So, yeah, again, depends on, like, is your emphasis more on the directionality of the evolution, or, like, let's say you're looking at transmission, or is what you want to communicate more of, like, oh, I'm sampling from, let's say, different parts of my country, my main aim of looking at this is like, do we have samples from the eastern and the western part more likely clustering independently, or do we have mixed sampling? Or do I, like, if I collected these samples within a year, do I want to check if, I'm seeing some temporal pattern, like I find January somewhere, February, we are seeing that, or like we have different branches which are evolving independently and things like that. So again, depending on what it is that you actually want to get out of it.
Right.
Hello, James.
Yes.
I think I agree with you. For me, it depends on what question you seek to answer. And that's what it, because some questions can be answered by unrooted tree. And even if you have them. So I think it depends on what question you want to answer. That will determine where you go to, whether it's rooted or unrooted. But for me, I personally have a bias for rooted tree for many reasons. Thank you. And I will know what. So for me, I will not use a midpoint tree. I would rather put a not too far genome in my tree file, I mean, in the analysis before I start. And then I know this is Salmonella, for example. It is this far from E. coli, and I expect Salmonella to be at this position, and then I can take my tree from there up. Thank you.
Good. I'm really glad for the question, because it highlights some of the things you've said, like it depends on the pathogen. It depends on also what you want to communicate. And also just, saying that, there are different ways of doing this. And well, there are also like personal opinions, like, one this big phylogeneticist to say is like, you only use a circular tree if you have something to hide. Again, that is his own personal opinion. Circular trees are nice to use. So I don't think it's only like when you have something to hide. But anyway, let's move on. So bootstrapping, again, using this resampling technique to determine sampling error. So ideally, what this communicates is how reliable are my branches or the cluster. So the way I typically read this is saying that, let's say if I do let's assume that these numbers here represent proportion out of 100%. So at this point, you're saying that my samples on the right cluster 90% of the time to the exclusion of whatever is on the left, and the same here, 65% of the time, these samples cluster together to the exclusion of whatever is on the left side of that branch. And generally in the field, bootstrap of 70% and above is considered reasonable. But then again, you'll note in the field that, some put a higher cutoff depending on the data, again, that you're analyzing. So if there are more recent samples, you want a higher bootstrap value to be able to really delineate samples. And again, depending on, the quality of the genome, etc. So this will determine what bootstrap cutoff value that you use. But of course, if you want to have strong conclusions about your data set, like, oh yeah, these samples cluster together, then, you need to implement strict bootstrap cutoff. And the way this is done, typically just think of, you have data set A, B, and C, and you have the whole genome, the original data, and then you resample different positions. So you have a genome length that is 4 bases, and you randomly pick each of these positions. Some of them, remember, will be duplicated. So for example, we have position 1 that has AGG, position 2, CGG, we have position 3, TSC, position 4, TTC. And in replicate 1, we pick position 1, as you can see from the original data set. We pick position 2, pick position 3, but then on the 4th position here in this replicate, we actually pick position 2 again. And then we build the tree, and we look at how it looks. And then replication 3, you can see, now we start with what was position 2 here. We've done our replicate. And in all these replicates, we summarize the tree and say, how many times, for example, do we see B and C together? See? Like how many times do they cluster together to the exclusion of whatever is on the left? Because as you can see in this replicate 1 and 2, B and C cluster together, and in replicate 3, A and B cluster together. So in the end, we assess how many times that happened. So in the last 5 minutes, we ask ourselves, so what can we infer from a tree? We can infer different things. For example, host switches, how a pathogen moves from one host to the next. Spatial movement, how a pathogen moves from one location to the next. Transmission events, from one host again to the next, or from one individual to the next. When a novel lineage emerged, and also when novel mutations occurred. So here I have a sample of 4 trees, A, B, C, and D. All of these are from the same set of pathogens, but you'll see that there's sort of like different clustering patterns. So some of you might be familiar with this figure. So this was sort of like trying to look at the origins of SARS-CoV-2. So you see here SARS-CoV-2 clustered together with this monkey. Was it rat? I think this is a rat or a mouse. I forget now what this was. But you can see, with R. malaius here. And in B, we can see SARS-CoV-2 clustering with this rat-G sequence. And in C, SARS-CoV-2 clustered together with the pangolin sequence. And in D here, they cluster together with, they share most reason common. So rat-G and RMYN02 share most reason common ancestor, and it's only their most reason common ancestor that they share with the SARS-CoV-2 sequences. So trees inferred from different parts of the genome or different genes, showing sort of like different topologies. And can anybody quickly tell me what we can infer from these 4 different trees from different genes? Any quick responses? All right, so
Sorry.
this is typically, yes, Rotimi?
So these are the same set of genomes, right? Or
So
different set of genomes?
these are the same pathogen, these are the same samples, but the trees are from different genes, so they're from different parts of the genome.
Oh.
So if I'll give you an idea, so this simply means that the different parts of the genomes have different evolutionary histories, and this is typically a clear indication of recombination going on in the data set. So the other thing that you might be able to infer is pathogen transmission. So here, looking at the pandemic H1N1 2009 tree, again, looking at, the origins in Mexico at the most recent common ancestor here. Again, the route pointing to Mexico, but then you can see introductions in the same city at different points in time. You can also see probably different variants. You can see New York here on this branch here, as well as New York on this other branch here. So sort of giving you an idea of multiple introductions within the same location. Also thinking about pathogen transmission. So again, I have this tree where I have in red samples from Kenya. I think this is probably one of my data sets that I worked on many years ago. And we have samples from different parts of Africa. And for example, here we're asking the question, where did these samples in Mali, which are in orange, where could they have originated from? So if you look at the orange ones, they cluster together with the samples from South Africa here, but then you also see Zambia, right? And you also see Gambia in blue here. But then they share most recent common ancestor with samples from Kenya. Again, you could say all these come from Kenya and then, spread to these other countries. But then again, you can see the Malian sequences cluster with Gambia. So you'd say like, well, those are, West Africa. So maybe, it makes sense that they cluster together. But then you have to think about oversampling. So in this case, Kenya was oversampled. So in such a case, everything seems to originate from Kenya. So just being aware that, as you build your tree, things like oversampling of any given location can interfere with your inference of transmission. Also looking at pathogen evolution. So if you're familiar with flu, you'll see clade replacement as well as divergence going on, coming from 3C to A, 3C to A1B, etc., going along the tree. And lastly, sort of what I did talk about previously, a cladogram. So this is a case where all the branch lengths terminate at the same point. So if somebody asks you, like, what can we say about the evolutionary distance between these highlighted taxa, it becomes quite difficult to say what distance that is, because then again, this is a cladogram rather than a phylogenetic tree. So the branch lengths really tell you very little about that evolutionary distance, right? It tells you about a given clustering pattern, but it tells you little about the evolutionary distance because, it is a cladogram. So lastly, as we finish, I know we are at the top of the hour. So some of the tree building pitfalls, one is not using enough background data. So as you build your tree, try to have sufficient background data or contextual data to make proper inference. Sometimes you might not have proper phylogenetic signal in the data set. Either you don't have sufficient variation to be able to differentiate between the samples. Also think about annotation errors on sequence alignments and dates. So for example, if you use the wrong date and you're trying to build a time-resolved tree, you'll end up with the wrong inference because of that. Not removing recombinants, that will definitely mess up your tree building. Also not removing sequencing errors. So again, going back to the idea of always just picking samples that pass QC, and always question odd results. Like you get something that, oh yeah, you've just discovered something new. Always ask yourself, like, what could I be missing here? And don't try to overinterpret gaps in your tree, like saying definitively, like, oh yeah, there's transmission from X to Y. Try to think about more of like this, try to get additional data that actually can fill in the gap, and do not overinterpret whatever it is that you cannot get or understand from your tree. So I think we'll stop here for today, unless there are any last minute question. I realize that we are 2 minutes over, but I'm happy to take any quick question, then use most of the office hours to go over this concept if there are any questions, and also look
Thank
at
you,
some tree
James.
visualization options that we have. So
Yeah.
yeah.
Thank you very much, James. Excellent talk, and thank you so much for all the effort. I just want to go back to last week with tree. My attempt to run RC typing failed, and presumably because I've exceeded my my credit, as it were. I'm trying to look for the right word now to use. Yeah, sort of. I've exceeded my cost limit. Cost threshold reached.
Well, so I talked to Kevin this morning, so that's not something we're expecting. So like I told you, yeah, I did see that in one of the workspaces, but we don't expect we don't expect that to happen. Let me see. This is your workspace, right? Yeah, I saw this error. So Kevin is looking at it because we don't, to be honest, we don't expect this to have happened. And considering that, your run cost is only 0.04 to the dollar. So Kevin is going to look at this and then let us know what has happened there.
All right, thank you. So I can't view any tree yet. But I was able to run the other one, the zip.
Yes, because you see,
workflow.
you've been able to, exactly. So you've been able to run this and it went through. And I think there are a couple of people who've run analysis after you. And remember, these are shared credits throughout all the training workspace. So if there was a limit specifically to your account, I would have expected the analysis for the others as well to fail. So is there anybody else who has seen this cost threshold reached in any of their runs today?
No, I haven't.
Okay. Yeah. So Kevin
Thank
is
you. I
looking
guess I
into
have to
it.
try again.
Yes, please. So again, if I might just have a quick second, the most failures that I've seen, for those that I was able to quickly look through their workspaces, just be cautious about, so let me just go back to the workspace data. Please ensure that, you know, when you put in workspace dot the key element here, ensure that it's sensitive to the case. So if yours is Staphylococcus xylosus and it's all lowercase, if you start with a capital S, it's going to fail because that element will not be recognized. So just check if that is the cause of the failure. Yes, Gumbo?
Hi, James. I was trying to create the TSV file. So I downloaded I selected those 28 samples and tried to create a TSV file, and I think I succeeded in creating that, but I failed to see it after trying to export it onto my workspace. I don't know how do you export it to the workspace?
So did you come to the data uploader and upload the samples?
Yes.
So
It said successfully, but then
no, so you can't. You needed to unzip this and upload them individually.
okay.
Yeah, this is not a single sample, but multiple. So you need to unzip it on your computer and then upload each of the samples individually. And then now you can upload. So after you do the upload, then you go next here, then you can now upload the TSV file. It should be fine. Does that make sense?
Okay, thanks.
All right, cool. You're welcome.
So, James, is Thursday our last session?
No, we are still meeting. So this is our last week, but we still have a session on Thursday.
Right. So this Thursday is the last one?
Yes, this Thursday is the last one. But we are hoping to meet the JIDC folks to talk more about the workspace credits. So that will be communicated in due course. But then hopefully if we have a bit more time on Thursday, we can go over any pending questions that you'd like to ask. I'm happy to spend a bit more time, even go back to whether there are questions on TheaProc, whether there are questions on RC typing, and maybe only have a few minutes to look at a few phylogenetic visualization methods that are available.
But then we'll still have access to the workspaces and everything after the training is over.
Yes,
that right?
that is the plan. That is the plan, to have access to these workspaces for a year from this course. So that should be 2026. So I think the meeting that we are supposed to have this week is just to iron out like how many credits are available,
right.
Then estimate like how much do we expect each of the participants to use, and if those credits are available.
I've started an analysis today. I uploaded some of my sequences and did the TheaProc workflow, but I haven't done any RC typing, and I'm not sure that I'll be done before Thursday, especially that I will need, I most likely need a different reference. It's a mixed staph aureus and other staphylococci. So I might need different references, and I'll need to look for these and upload them and all that.
Okay, so before then, so I think take it from me like, yes, you have 1 year from this week to be able to have your workspace. So
Right.
don't worry, don't be in a hurry.
Mhm.
But also I will need to, as promised, like write a short write-up on how to prepare those references. So
Right.
hopefully that,
Right.
yeah,
Right.
that will be helpful.
Thank
as well.
you. And if we have questions after Thursday, can we still email you, or you won't be available to help with any problems we run into?
So I'm going to ask Kevin about that, but I
Okay,
think,
okay.
yes, definitely email me if you have any questions. Yes, please do email me if you have any
And
questions. Yes.
if any publication comes out of the work, I will need to know how to cite so the workflow or JIDC, or I mean, what's the proper way to acknowledge the help we received and everything? Because I'll need to do that.
That's a good question. I mean, how to cite Terra, for example, that's easy. I think we can find ways to do that.
Okay.
I think
But also
I should.
JIDC, I think we'll need to acknowledge because they pay for
Claire
the
is
training.
here. Claire, do you have any insights on this?
Hi, so I'm Claire. I was working with JIDC on sort of testing and development on this side. That's a good question. And I guess that will have to be covered in the next JIDC catch-up meeting and things like that. To my knowledge, then acknowledging JIDC in the first instance, but I guess we can look at confirming and maybe creating some user guidelines that yeah, I'll bring. I've just got some notes. I'll bring that up in the next meeting.
Perfect. Yes.
Thank
Please.
you.
Yeah.
Thank you.
Yeah.
Okay, so thank you. Thank you.
All right. Any other last minute questions? Otherwise, we'll end today's, just stop the recording.